Yeah, thank you very much for the nice introduction. Also, many thanks to all the speakers we had before, because I think now it's really a challenge to add something new, because I think there was so much cool stuff already out there about open microscopy. So I just tried filling some of the holes that are left there. So actually right now, maybe it's the best proof that open source microscopy is useful, because I'm not in Jena right now, but I'm assembling a microscope together with some other people remotely. And yeah, so our mission in a way is making microscopy as simple as the tool, what you can see here, which is essentially a hammer. And I think my mission is like, I have to understand everything. So we build Storm, Sim, and all kinds of other things. And yeah, this is really fun. But it also addresses serious issues. So I think in many optics labs, you see this here. So on the left-hand side, you see a rendering I picked up from Twitter, and an actual setup, which I think is for some quantum entanglement. However, I couldn't really follow the path, so it's really becoming difficult to replicate it. So microscopes, in a way, behave similarly. So it's, I think somebody already has mentioned it today, it's a black box, and you cannot really add anything once you bought it. That's kind of a bad thing. And what we're trying to do is kind of bringing the two different fractions a bit closer together. So the users who are, well, really have to use these tools, and those like me who build it. And so how about just bring them together and create better tools that are actually useful? And luckily, there are tons of tools already out there. So I think if we just have a glimpse on the software community, there's already Fiji, of course, mentioned Imjoy, Cellpose, Micromanager, or the Zero Cost for Machine Learning. And recently, there's also some movement bringing these tools also to the hardware. So some don't even use any optics, like the diffuser cam. I think the open flexure is very famous, the Foldscope as well, even though that's not quite open source. But there's tons of tools that really can create an impact, because you can just multiply them, you can build them anywhere using 3D printers. And that's a bit what we are trying to do as well. The problem, however, is in many cases, software, if it's well written, so you just download it, install it, and then use it. So that's all fine. In hardware, that's a bit more challenging, because you have to download it, do the shopping, building, assembly, debugging, because it never really works in the first place. And then eventually you can use it. So I don't know, whoever has eventually built one of our systems, like these little tiny cubes here, or an open flexure, it takes some time to get used to it. And so what we're trying to do is a bit changing that, so that building hardware becomes a bit more off the shelf. And how do we do that? So first of all, of course, you have to make it super available by means of, so there are already tons of tools nowadays, which were not there like 10 years ago. I think Johannes already mentioned. So you can buy CNC machine parts pretty much off the shelf, or you just buy one of these machines that are in the range of a 3D printer in terms of price. You have 3D printers, you have nowadays also the PCB printers. And so all these things, even injection molding machines, they really support your idea of creating hardware devices now in a very standardized way. And in addition, you also have lots of different parts that are becoming cheap, and also nowadays quite nicely documented, because many people from different fields also contribute to the open hardware mission there. So you can, for example, replace very expensive cameras. So we did that with the Cellstorm, which was already nicely mentioned, thank you, with your cell phone camera, if you know how to process the data, because they always come with problems. And so there's already tons of different devices you can use for your research in order to create super resolution images. And so we created many different devices, where one, for example, was the Cellstorm. So I think I'm just skipping how a storm actually works, because I think we have heard that quite a few times today. But the problem is, of course, then in many cases that you use this bulky machine with a bulky camera. Well, in the first place, we just replaced the camera with a cell phone. And then you can eventually achieve a resolution in the range of 80 nanometer, which was already quite nice. But the problem is still, of course, you have the whole setup there. And in the first place, we thought, okay, how can we shrink it? And then we create this other thing called Cellstorm 2, which is a cell phone driven and also fully operated microscope. It's fully 3D printed, and it makes use of these photonic chips. So I'm not going too much into the details here. You can nowadays buy them off the shelf. And you can use it as a waveguide, where you then have a laser coupling into these little waveguides here. Also with an optical pickup unit you can get from your PlayStation, that can actually really nicely couple into single mode fibers for just €5. And then you excite whatever cell or fluorescent organism is sitting on top of this chip here. You image that with a very simple microscope, and then you can do super resolution imaging. For example, by just shivering the lens, you can excite different modes inside the chip, and then you can get this funny pattern which you can then use for example, machine learning driven super resolution. So we just tried one approach that's similar to the SURF from Ricardo Enrique's group, but purely running on the cell phone itself. So you ported it into a little Android app, and what you can see here is the membrane of some labeled E. coli, and the wide field image here. So this is real time as a screencast. You can see that the membrane eventually gets recovered. So this is in the field, so to say. So it's heavily vibrating as well. And another thing you can do with it is actually single molecular localization, where you for example, bring this whole device into a high safety lab where you eventually can never really bring this device out anymore. And then you prepare your sample. In this case, it was active SARS-CoV-2 viruses already quite some time ago, which we labeled, and then you can do storm on the data here. So you see the bright field response and the storm response all done on the cell phone. Which by the time I found quite impressive, because the virus was essentially right in front of me. So, but that's just one thing. Sometimes you're asking yourself, okay, I'm in the field, I need a microscope, what should I do? And together with Vittorio, who also contributed to the field guide for 3D printing, we had this idea to eventually have a small device that's nothing more but just a small microcontroller. So you can get this for €5. It's an ESP32 camera. And if you just extend the tube length between the objective lens and the camera, you already have a microscope. And it has a small LED. And if you guide the light such that it becomes an illumination source, you're already done and you have a microscope. We have a small 3D printed housing. And then we thought, okay, so what can we do with it? Of course, well, this is for detecting, for example, parasites, but there's one more thing you could do for it. In marine biology, many people are interested how biofilm is forming. And so we thought, why couldn't we just build a small microscope that can be waterproof, can be cheap, it can capture long-term series of images underwater, and how can we do that? And then we did a little survey on Twitter asking people who has access to this jar. Because we thought, okay, I think you can just do it with PVC pipes, but marmalade jars, they are available everywhere. And so this essentially is available in most of the countries on Earth. And together with this, we built a small waterproof microscope. It's now called the Anglerfish because the illumination is guided through a light guide from audio technology. And so this whole thing costs in the range of like €20. And we also recently published a small assembly guide on YouTube. And this thing is just there for capturing biofilm growth underwater. So this is the some time-lapse you can see here. And it also features a small voice call motor-driven lens you can find in cell phones in order to get focusing capabilities. And also this whole thing is open source. I can provide you with the links later. Yeah, and then briefly talking about what we are working on really heavily recently is the modular system, and to motivate that a little bit. So why is it important to have a modular system? So I started with a background in industry. When I did my apprenticeship as an electrician, we had to design electrical circuits many times, and usually it takes a really long time to design it, to build it, to test it. And then once I was done, there was this little thing, the Arduino. And I think most of you know it. It's an open source microcontroller. And this is really cool because since it's open source, it has a large community where many people contribute with open source code. And you can create arbitrarily complicated projects in research, industry, science, at home. And this is really cool. And we thought when I was starting my PhD in Rainer Heinzmann's lab, we should somehow copy this idea to microscopy, because microscopy itself is inherently modular. So if you observe the different relation between the lenses and light source, objects and camera, they always follow by the distance of one focal length. And if you then just draw boxes around it, you're already there. That's called UC2 now. And so it's really simple. And we created many different setups, which I think is also important to discuss later on, because there's so much waste and we have to think about recycling it in a very nice way now. And right now we ended up with a kind of a library for hardware modules. So you have a module, for example, for laser, for beam splitters. I have one here that's a motorized beam splitter for fluorescent microscopy, which I just exchanged here in the setup here. And with this, you can then just build whatever microscope you have in your mind. For example, an inverted microscope with phase contrast, which you can fit many of those in an incubator. Or you can also build multicolor fluorescent microscopes, as you can see it here, to, for example, observe the entry of virus or bacteria into a cell. Or you can also, I think this has been shown many times today, do super resolution using single molecular localization. And the cool thing is, since it's open source, I can just openly talk about it. And there are many people really interested in collaborating. So you learn on both ends. So those who build learn how to build, and I learn what's the process in the end, how to get the biology right. And then we also tried to do some stuff regarding the structured illumination microscopy. So Harun Wang is a really talented PhD student with us, and he was able to build a small module that you just snap in your ordinary fluorescent microscope, and then you have super resolution, mostly along Z, so you get better optical sectioning here. You can also do OCT. So many people always ask, plastic is not really stable. We tried to disprove that by means of you can create stable interference signal to eventually also recover 3D information. So this is very, very new. So we don't have reconstructed data yet. And one other case where this modular system is very, very powerful is when you want to create new systems that you just want to create very, very fast. So recently I joined Manu Prakash's lab in Stanford for a short visit, and there we were asking ourselves the question, how could we image plankton? And plankton is very diverse. You can see that everywhere on Earth, there's even a mathematical formula to define the structure of what you see here. We of course thought about, okay, let's try doing it with some kind of a light sheet system so that we can use the autofluorescent signal and then capture the 3D shape of the particle. And so there are different systems, of course, the SPIM, and then something where you blow the sample into the focus, or some vertical system here. But we want to have one more information. So not only the autofluorescence, but also the scattering by means of, for example, holographic detection. So we ended up with this here. We call it HoLiSheet because it's a holographic light sheet. And so the idea here is you have a rotating capillary, and then at the same time you have a pump pumping your sample volume through the focus. So that means your sample makes a spiral through a light sheet, and at the same time you also collect the holography. In this time it was a inline holographical signal with 2 different cameras. And so this here is a simulation. In reality, it looks a little bit different. So what you see here, for example, is one algae. These are some beads I quickly labeled, and this is the rotating capillary. So this whole system kind of works, and we also tried that, tested that in the field. And so the reconstruction of the fluorescence here already looks quite okayish. However, the holographical signal, I think some more computational or algorithm challenges are to be solved there. And the cool bit here was then the rapid prototyping aspect was nicely used, and in the end we were just able to derive the whole system by means of, okay, it was important to use, for example, pipette tips as bearings because they seal and they are really straight and cheap. And with this, you were then able, in combination with the small motor and the FEP tube, to create this little device which did exactly the same, but on a standard microscope. So we did the rapid prototyping in our system, and then once we were done, we just translated that into this little device, which now can be used by anyone. It's also open source and documented in GitHub. And then, yeah, this is really nice case how to use this thing. And the last note, we also made use of another open source system, which is an open source pipetting robot. It's called OpenTrans. And we were asking ourselves the question, how could we eventually create a fully automated laboratory system where the microscope and the robot can interact the way that you create better protocols. And so we teamed up with people like Richard Bowman and Wei Young from OpenFlexure and Enjoy. And so we created this feedback loop where you have microscopy and biology somewhat digital, and then create a feedback loop so that the next round of the next biological protocol may work a little better. And so what we did, since I have roughly no idea about biology, we just used some yeast cells and added different solutions of sugar and peptone in order to increase the growth rate. And so the little microscope, what you see here, is then doing automatic imaging, and then finally also analyze the highest growth rate there. Yeah, and so finally, I think Johannes nicely phrased it already, so I don't need to talk about that too much. Openness is something not only important in terms of reproducibility, but also a really nice aspect for your, or at least for my work. So I make heavily use of available tools. For example, recently I had to review a paper in this journal of open source software. It's about this InSwitch software that's a controlling software in Python. And so now we are able to use it together with some code we derived from other sources. So it really speeds up our research, because I never really start coding from scratch. I can always take code, of course, always taking into account that the license has to be right. And also others can step on that. So for example, recently there were some publication taking our cubes and putting that into a quantum experiment. And many people built their own microscopes because now I think they kind of lose their fear to just do the first step. Or another way where there's some people in Paris creating an XY stage. We just adapted that to our system, and then it quickly started working. Yeah, and together with all that, I am really proud to have so many people all over the globe using our stuff. Just 2 weeks ago, we had the first people in Madagascar using our tools here, and they, I think, for the first time, used the microscopes to detect some living fly, which was amazing. Yeah, and almost done, but there's still one more thing. So the problem, of course, is the scaling aspect. So how could we scale the open hardware bits? And so we tried many different ways, and finally we came up with the idea to found a company that just translates the open source the way that people eventually can buy it, because our research institute is in a way open, but of course they cannot just produce things and sell it. So that's not in their scope. And so we try this critical aspect so that we maintain the accessibility and adaptability and the open source, but still try to bring that to the people by means of a company. So we are really curious how this will work. It's a lot of work. I think similar to a large scientific project, but yeah, I'm curious how this turns out in the end. So to summarize, yeah, did we make microscopy as an off-the-shelf tool? I hope yes. The community aspect is definitely important, and we have to maintain that. Distributing hardware, that's something on our list, and something that's not really on there yet, but I'm super keen to do that, is like connecting all the different instances of microscopes. So not only OpenUC2, but also OpenFlexure or the MyCube, because I think it would be really nice to give others, not only us scientists, the ability to study or to visualize what we see every day. Because yeah, just having plankton on a big screen is already very amazing. So that's definitely something I would like to do in the future. And with this, I am a bit out of time, very sorry. Would like to thank you for listening, and yeah, I'm very excited for the next discussion.